Right, component 2, question 3, here we go. Okay, it gives you a lovely diagram of the pollen grain development. If you remember, we've done this practical with the ready-made slides because the ones you've stained yourselves, they never show up the nuclei the same way. So from the ready-made slides, you would have seen all these. Now, the questions is literally asking you what phases and what stages, identify which stage of meiosis can you see. What are the hints that it gives you? It tells you here homologous pair. So two pairs of chromosomes pulled to opposite poles. So pulled to opposite poles, that has to be your anaphase. And because they're still in homologous pairs rather than the individual chromatids being pulled away from each other, you've got anaphase 1 for this one. Reformation of the nuclear membranes, that has to be a telophase. Definition of telophase is nuclear membranes being reformed. And then it tells you around the gametic nuclei. So the gametes are already formed. We're already at the stage where we've got the haploid information divided even further. So at that point, we've got telophase 2. And the third one here, it gives you, again, it gives you enough information. It tells you arranged across the equator, so that alignment across the equator and spindle fibers attaching, that is metaphase. Now the question is it metaphase 1 or 2? Chromosomes forming the bivalents. So there's still the homologous pairs, bivalents lining up side by side. That is your metaphase 1. Right, and this question is a bit sneaky. It goes back to the core concepts. I remember doing this with you and drawing up the animal cells as a dividing to show you the cleavage furrows that form and making it clear that as a plant cell divides, you've got the plate forming in the middle rather than having a cleavage furrow. The differences in spindle fibers and how they're in animal cells being packaged away again into centrioles, but in plant cells, those spindle fibers, they're just staying, they're just there. So 2 ways in which meiosis in animal cells is different than in plant cells. Mark schemes tells you the organization of spindle fibers into centrioles in animals, and the cleavage furrow that you'd get, the cleavage into new cell membranes. It would have, and the Mark schema does not say anything about the plates instead of the cleavage furrows, but that is the idea here, and the converse argument the other way around is absolutely right. Good. Then you have your different stages in spermatogenesis compared to the diagram that you've just had. In spermatogenesis, what would be the equivalent of diagram A? So if you go up to diagram A, you've got a prophase, you've got a cell, sorry, not even in prophase, you have got a cell that is still undergoing mitosis here. That's mitosis and replacing itself. We're talking about the equivalent being spermatogonia, the germinal cell line here. And then it's telling you F, what is the equivalent of in sperm development, what is F? You have just had telophase 1. So you've got the 2 nuclear membranes, you've got cytokinesis even, so you've got a cell membrane in between, which means you've now got haploid information. Double of each, but you've got haploid information. The actual information itself has been halved, the information quality, if that makes sense, which means you've got the secondary spermatocytes here, and then it's asking you about K, where you have got differentiated already. Have a look, it's already, you have got the haploid information halved into the individual nuclei. You've literally got the gametes made, so spermatozoa at this point. So spermatogonia, secondary spermatocytes, and the spermatids stroke spermatozoa. And wait, going back down to the actual question here. In mitosis, so the question is now flicking from meiosis over to mitosis. The cells formed during cytokinesis would re-enter the cell cycle. So you've got a normal average cell that just does half the genetic information, divide the cell, double it all up again, double the cytoplasm, you know, the normal cell cycle. Why can this not happen once you have got sperm cells? You've got spermiation that has taken place. You've got the differentiation part where the Golgi body is changing into your acrosome, where your mitochondria line in the midpiece, and so on. So where you've got spindle as well in the midpiece as well. So we've got that differentiation, and what the epididymis does as well later on, the drying out. Once it has been differentiated, once it's fully differentiated, it can no longer re-enter the cell cycle. Same thing for the pollen grains. Once they've gone through the formation of the outer fairly drought resistant, drying out resistant layer, the intine and the exine bits. Once you've got that, it cannot re-enter the cell cycle. Here we go. Maths. Don't we love a little bit of maths? So what is it that you're going to have to think through? It's telling you, you're looking at lilies and the diploid number of chromosomes, that is 12. Diploid number, not haploid, the diploid number. So the number of combinations of chromosomes in the gametes, how much information goes into your gametes? You automatically should click, that's when we're at haploid. In this species can differ due to independent assortment, and it can be calculated 2 to the power of n, where n is the haploid number. 12 was your diploid, your haploid number is 6. Your maths is literally 2 to the power of 6. And if you cannot do that with the calculator, then it is 2 times 2 times 2 times 2 times 2 times 2. Did I do it the right time? Right? No, the right number. So 6 times. And you should get to your 64 with this one here. Okay. Various other wrong options give, okay, you can get 1 mark for kind of being halfway there, for not clicking in haploid and diploid. At least you get yourself some marks. Remember to show you're working, even if it goes wrong. The stages of meiosis where independent assortment can happen. So that is literally where you've got the lining up along the equator, and it could go either way from there. That initially the mom's copy of chromosome or the dad's copy of the chromosome would go into cell A or cell B, but then actually second time around again, where in metaphase 1 and in metaphase 2, you have got that lining up along the equator, and it can then go either way. So metaphase 1 and metaphase 2 both need to be there. Where are we going there? Here we go. Apart from mutation, name one other source of genetic variation during sexual reproduction. During sexual reproduction, not during prophase. So this is not allowing you the chance to put down that you could have crossing over events happening. It's asking you specifically during sexual reproduction, something that does not result from meiosis. Okay, so you can't have the prophase 1 and the crossing over event. That is not a must, not an option here. This is a random which sperm happens to win the race, which egg, which pollen, which ova happens to be actually doing the fertilization. So a random fertilization and or describing that. So if you were to say you're not quite sure which one of the however many million sperm released or whatever is going to make it. And the last one, not least, but that's your 4 marker. This one was a little bit longer. Let me kind of think through myself what that one here was. Apple trees produce pollen grains that are not self-compatible. Oh yeah, that was the question where you've got the development of stigmas depending on air temperatures. Okay, so the female parts triggered by air temperatures. However, some other species, you've got it triggered by increasing day length. I'm recording this on the 21st of December. Okay, here we go. It goes only uphill from here. So the 2 different definitions here, like some flowering plants develop stigmas based on warmer air temperatures, and some are doing the day length thing. So a light trigger. So clicking in that there are 2 different varieties here. Cross-pollination often occurs between a variety which is flowering triggered by longer day length and one that is triggered by warmer temperatures. Using this information, conclude why commercial apple growers, and it says at the start, the different variety pollination gives you cross-pollination is more beneficial in that context. Using this information, why are UK growers concerned about climate change and why it could give you a lower yield? So if you've got pollen grains that are self-incompatible, you do not want self-pollination. You want cross-pollination to get a higher apple yield. The key thing to click in here is that global warming will affect whichever flowers that are triggered by the warmer air temperatures. So whichever carbon dioxide in the atmosphere, methane in the atmosphere will increase air temperatures, and therefore the varieties that are triggered by warmer air temperatures will be triggered earlier in the year because it gets warmer earlier. However, the day length, I'm going to remind you of something you did in year 7 when you did planet Earth circling around the sun and the tilt of the axis, blah, blah, blah. The day length is not affected by global warming. That is simple physics and planet Earth on its axis being around the planet where it is and gradually getting longer and longer light hours based on that planetary movement. That is not impacted on by the climate change. So that these varieties will stick to their, okay, we've had longer day length timings, therefore we are flowering at this point in the point of time in springtime rather than whatever the warmer air temperature said. Therefore, those varieties are not ready at the same time. Going over to the mark scheme because that's the language we need to be using in the end. So, because of global warming, we're getting higher air temperatures. Those trees that flower earlier due to the those flowers that are relying on the air temperatures could flower earlier. Also, oh yeah, the pollinating insects, you could get that one into your argument as well. If you get insects earlier, because other species of plants might be around earlier, and therefore insects come out of their larval and pupal stages earlier, that can go into this argument here. However, the day length is not affected by climate change. That's just a planetary thing. Therefore, cross-pollination cannot take place because the stigmas are not ready at the same time. Therefore, less pollination overall, and therefore the crop yield will be lower. Quite a bit of an argument. I did let you get away with quite a lot of alternative arguments, but you need to hit the gist of this here for me. Right, that was question 3.